Behind every pioneering idea, method, and device is a fellow human or humans, a trailblazer who is daring enough to ask the questions that push the boundaries and make the impossible possible. I'm Sharon Kedar, co-founder of North Pond Ventures, a multi-billion-dollar science-driven venture capital firm, and the host of Innovate and Elevate. In each episode, we'll have candid, in-depth conversations with top doctors, scientists, and innovators about leading-edge discoveries and how they impact our lives. Season 1 focuses on women's health with the aim of helping women lead our healthiest lives. You'll hear from leading experts such as Dr. Catherine Rexroad, Division Chief, Women's Health at Harvard's Brigham Hospital. It's time for all of us to innovate and elevate. Welcome, Dr. Don Ingber, to the podcast. Dr. Ingber is the founding director of the Wyss Institute for Biologically Inspired Engineering at Harvard University. The Wyss Institute was founded 15 years ago in 2009. Dr. Ingber is the Judah Folkman Professor of Vascular Biology at Harvard Medical School and Boston Children's Hospital. Dr. Ingber is the Hans-Jörg Wyss Professor of Bio-Inspired Engineering at the Harvard School of Engineering and Applied Sciences. Dr. Ingber's human organ-on-a-chip technology has been honored as one of the top 10 emerging technologies by the World Economic Forum, Design of the Year by the London Design Museum, and acquired by the Museum of Modern Art, MoMA, in New York City for its permanent collection. Dr. Ingber is scientific founder, board member, and scientific advisory board chair at Emulate Inc., the leading manufacturer of organ chip systems. Dr. Ingber, welcome to the podcast.
Thank you.
Don, it's been such a pleasure to know you. I think it's been 5 years knowing each other both as fellow board directors on Emulate Inc. and through our unique partnership with North Pond Labs and the Wyss Institute. I wondered if we could have the viewer and listener get to know you a little bit better. You have appointments through surgery and engineering. You founded the most innovative institute at Harvard, and your inventions are in art museums. You could have chosen many paths as a scientist and engineer. Can you take us back to your younger self and just tell us a little bit about how you got here?
I grew up on Long Island in New York, middle class. My parents didn't go to college. I went to public school. I was lucky enough to get into Yale as an undergrad. I was strong in science and math, but when I got to Yale, it was this incredible liberal arts institution, and I kind of held back a little on the science and took courses in everything I possibly could and got really in love with just learning. And I started research there when I was, I guess, a junior, but it was also the same time that I took an art class in sculpture because it was called three-dimensional design, and I was doing work in molecular biology where everything was at the molecular level, three-dimensional design. And in this art course, I learned about a way to build things called tensegrity that comes out of the Buckminster Fuller world of architecture, sticks and strings that don't touch, that hold themselves open. And I learned about that the same day I was culturing cells in a cancer lab where cells were changing shape, and the professor in this course had used one of these models and it changed shape the same way. And I basically realized that cells must be built this way, and that sort of took me on a path towards pursuing this idea, which I just thought was obvious. But then I got into medical school and graduate school at Yale, and I started pursuing this even further because I could see it could explain things I couldn't. And so that got me on a path to sort of thinking about structure in biology and mechanics in biology. And I got involved in cancer research, and that eventually brought me to Harvard because I did a postdoc with Judah Folkman, who at that point had the idea that you could treat cancer not by killing cancer cells, but by starving the tumor of blood vessels, inhibiting capillary blood vessel growth, known as angiogenesis. And that was very physical in nature. He also had found that the shape of the cell controls its growth. I won't go into everything in my path, but the point was I just followed the path of least resistance, like where my passions lied and where I thought I could see things other people couldn't see. and finding the right place to be able to pursue them. That brought me to Harvard. And, long story short, it brought me into discovering that one of the first drugs to go to the clinical trials to treat cancer by inhibiting angiogenesis. It brought me to work with other faculty like Bob Langer, who pioneered so many entrepreneurship in science, basically. And I got involved in startups and tissue engineering. I wanted to pursue my ideas that physical forces, mechanical forces can be as important as chemicals and genes, which came out of this sculpture class in tensegrity sculptures. And so I collaborated with engineers and had to learn engineering in the process. And all of that kind of took me on the path where I am today. If you just follow your passions, you'll end up where you want to be. And that's what I've done.
When you were in college, what sparked taking this art class that has really transformed your journey? Was it interest, random, both?
Well, one thing I'll say is that, when I was a kid, my hero was actually Thomas Edison and not Einstein. So the inventing part of this and physical building things was always part of it for me. But I took that art class because I saw kids walking around the campus where they were holding things that visually, to me, looked like biology. They looked like the structure of a virus, so they looked like the structure of the molecule, but they were beautiful. And to me, that meant that they were doing something that had the essence of how nature builds. And when I asked them, what's the name of the course, they said three-dimensional design, which, as I said, is kind of how we were learning about molecular biology in those days. The shape of the DNA, the helix, is what stores information. You could make it out of other chemicals, and if they match that way, they do the same thing. So it's really the shape and, lock and key and enzyme substrates that they bind together like a lock. It's the shapes. And so that's what brought me to that.
May I ask you to just explain to all of the viewers and listeners, what is organ-on-a-chip technology? What's your big idea and vision for its impact on the world?
Yeah, so that little device that Sharon held up, organ-on-a-chip, it's a device, as you can see, it's about the size of a computer memory stick. It's made out of optically clear rubber-like material. We used computer microchip manufacturing methods to create it originally. And what we do is we create two hollow channels that are parallel to one another. You could think of like two tunnels that you drive through, and the wall in between has holes in it so things can pass back and forth, small things. And we use that to recreate the structure and function of, believe it or not, human organs. So for example, for the lung, for the air sac of the lung, we have the lining cells that line your air sac on one wall of that channel. And on the other side of the porous membrane, we have the capillary blood vessel cells. But what's cool is it's made out of a flexible material, and we have little chambers that we can apply suction to that can stretch and relax. So we can mimic breathing motions, and we could put air in the top channel over the air sac cells, like in your body, and we could put flowing fluids like a blood substitute through the blood vessel channel. And when we do that, we actually recreate the structure and function. We line it with living human cells of an organ. And the same little device can be used to model the intestine by using cells of the intestine lining and blood vessel and giving peristaltic-like motions, or of the skin, or of the kidney, or of the bone marrow. And so we are able to actually recapitulate the structure and function of living organs this way. And the goal is really to replace animal testing, to be able to have personalized models so that you could have like your lung on a chip and test a drug, or your lung cancer on a chip and test the drug. Will it work against the cancer, but which one produces less toxicity in your liver and kidney? And really be able to shortcut, accelerate the drug development process faster, cheaper, more effective, safer. Because right now, animal models take a lot of time, a lot of money, and they, more often than not, do not predict what happens when you get to a human clinical trial. And that was really kind of what triggered the development of that.
I read an article with you commenting about how an organ chip is much better than an animal. For instance, if you're taking a drug 3 times a day versus once a day, if you're testing on an animal, the animal likely doesn't have the disease that you're testing for. So can you talk about some of the facets that you think are better in organ chips than animals?
Oh, absolutely. That is absolutely true. First of all, animals are often inbred, have very similar genetic makeup, so that you get very similar results, which makes it easier to interpret. But that's not the way humans are. And humans are incredibly diverse, right? And so the other thing is that the cells come from patients. So we could say test chips, Caucasians versus Afro-American versus Hispanic. We've done this with vagina chips, for example, where drugs are going to be tested with women in Africa versus the United States. You can do it with adult versus child, man versus woman. There's real problems in clinical trials that women are never as well represented as men. With these chips, you could do 50-50 sorts of testing. So lots of things you could do in these chips that are better than animals.
The technology is truly incredible. What do you think the biggest barrier is to adoption?
I think it's overcoming the natural human aversity to risk-taking in the workplace. Because one of the places where Emulate has shown that chips are incredibly effective is at modeling liver toxicity, the injury to the liver by drugs, which is a very common place for drugs to fail in the clinic or after they are approved to have problems in the clinic. These sorts of chips can be used to inform when you get conflicting results, which pharmaceutical industry often gets during the development process when they use different animal models. I think it's really the risk. They're used to dealing with animal models. Everyone else deals with animal models. So you as an individual in the trenches, if you're a toxicologist, have to be willing to take the risks to say, I'm putting my bet in this new technology. And that's true with any new technology when it's taken in. It's very hard to get those first users to make that jump.
When I think about innovation, I do agree with you. I think it usually takes a catalyst, and you almost need someone to step forward so that others basically must follow suit or come across a bit like more archaic. But that vicious cycle of lack of innovation turning to innovation and adoption, it's a fascinating journey, and it'll be interesting to see what catalyzes the adoption.
For the last 15 years, I've been at meetings with the FDA, with European equivalent agencies, with pharmaceutical biotech. Everybody agrees that animal models are suboptimal and have real problems. Everybody agrees that there need to be alternatives. And now that there are alternatives emerging, the question is who's going to take that step forward to take the risk. I think the first company that starts getting drugs through faster at a lower cost, when that happens, everybody will shift. But you need that one first company to go through, get drug approval, and be open that it's used this process.
Can we talk about an area that you've been a trailblazer? So women's health is an area that the Wyss Institute has focused on. Thank you for inviting me a year ago to do a panel on the topic. I think the White House did their first ever initiative a couple months ago, Women's Health Initiative. Renee Wegersine from ARPA-H invited me to the launch of that. And, you know, I was struck by the massive underinvestment in women's health. Dr. Catherine Rexroad sort of opened my eyes to a lot of that. I know she's a visiting scholar at the Wyss. You already have organ models for, I think it's the vagina, the cervix, and the fallopian tube. So can you talk about how those models might transform women's health and just your broader perspective on organ chips and women's health?
Sure. Gates Foundation came to us. They've been funding work we were doing in intestine with malnutrition in low-resource nations, which is going really well. And they are trying to develop a therapeutic that would be low cost, that could be broadly available for bacterial vaginosis in women. Now, bacterial vaginosis happens all over, not thought to be that serious in the United States, for example, but it's a major cause of preterm labor, loss of the fetus, and increased infection rates, all kinds of problems, particularly, in these low resource nations. And so they were looking for effectively a probiotic therapeutic. They call live biotherapeutic product, LBPs now, which are living microbes from the microbiome of the normal vagina that came out of research from a number of labs in the field that suggests that there's a healthy microbiome and an unhealthy microbiome, and that if you could flip it from unhealthy to healthy by delivering the good microbes, that you may be able to prevent this condition. But you can't test this in animals because animals have different microbiome than human. Mice don't have a menstrual cycle. And so they asked us, could we build chips that would model the vagina and cervix? If so, could we culture microbiome on it? Because we had cultured microbiome in intestine. And if so, could we test these products with the idea that they're going to a clinical trial, but if we did this in parallel, if this really predicts, then we could optimize, develop better and better versions of it. And so very simply, we developed a vagina and cervix chip. We can grow microbiome on it. If we put the bad microbiome on it, we see injury, inflammation, all the things you see in vivo. If you put the good microbiome on it, looks healthy, they live, they're in balance. And you could flip it in terms of suppressing the inflammatory response if you add the good microbiome to the bad microbiome. And now we're working with them with this live biotherapeutic product that's going to clinical trials in Africa and at Mass General in Boston. And, we are basically trying to use that to optimize, improve the formulation. So that's how it started. We've literally put human sperm in these chips, and you could see sperm motility. You could see how bacterial vaginosis affects that. We just got another Gates grant to study non-hormonal contraceptives using these much more natural environments. Part of that, we're developing a fallopian tube chip. We're just beginning. That looks interesting. They allow one to study processes that are incredibly hard to study in vivo, incredibly complex. For example, the obstetrician gynecologists that collaborate with us, it's like they have all these questions like, what's the role of the microbiome? What's the role of pH? What's the role of oxygen? You can't control these parameters in a human independently, or, you can never study human without a microbiome. And so we can study which one of these contributes.
As you're talking, I think about the data gap and where organ chips can help solve it. Just the reality that women were not required to be part of clinical trials until 1993, which is 70 years, I think, after women were given the right to vote. It's just sort of mind-boggling when you think about it. And then when you talk about things that I was not aware of, like mice not having a menstrual cycle, how do you even catch up with the data gap? And it seems like organ chips could be a huge part of the answer.
Yeah, absolutely. We model hormonal cycling during the menstrual cycle on the chips, and we see changes in the cervical chip that looks just like in vivo. We take little samples like a clinician does to look where you are in the cycle. We get the same sorts of readings. We've looked at how hormones as well as bacterial infection affect the cervix and the structure of the cervix, which could be involved in preterm birth and what leads to preterm labor. You could see breakdown of the wall of these chips, just like you see breakdown in the cervix that loosens and causes preterm birth. We see cervical plug formation, mucus production. And yes, you can get cells from premenopausal, postmenopausal women, explore what's different. You could, look for non-hormonal ways to modulate it. So women are not taking these incredibly potent hormones that have other potential deleterious side effects. So yeah, it opens up this whole palette to paint with in terms of both discovery and therapeutic intervention, as well as diagnostic intervention, being able to diagnose. And it can be personalized potentially. It's cost, right? But I think of personalized two ways. You could think of it like your, lung on a chip or whatever, but it also could be chips that represent a genetic subpopulation, right? Or a very similar group of people in terms of background, age, sex, ethnicity, et cetera. And that's important because most drugs now, drug companies spend a huge amount of money, tens of millions or more to get to the clinic. Then they do an expensive clinical trial and they almost always fail. And what they then do is basically do statistical analysis to say, is there a subgroup that responded a little better? And if they find it, a couple of years later, they'll do a targeted study. And if they're lucky, they'll get approved for a very narrow application. This age group, sex, this disease, manifestation, these symptoms, whatever. If you could start in the beginning by developing drugs for, identify 50 Hispanic women who have the same condition, let's say, chronic obstructive pulmonary disease, and they're ultra sensitive to the smog, and you get those 50 women and you develop a drug that works with all those women, and you can get their other organ chips to look for minimizing toxicity, then you use those women for the clinical trial. And that would be faster and cheaper and more likely to succeed, in my view.
I know that you often take a problem and approach it from how do we solve, a disease or a problem. And so there's so many ideas running through my head, but, you take something like menopause, where women are basically lab experiments today, and, you use this chip instead. That's probably preferable.
There's no better model than human. I mean, in the end, that's what we really need.
You're willing to be creative. You're willing to take risks. You're willing to try. And it feels like with the topic of today, women's health, we just have to try. And it feels like there's just so much low-hanging fruit for, a brilliant scientist such as yourself. Why do you think it's an under-invested area?
I don't know the answer to that, but I think some of it is historical, in the sense that people tend to work on what they've worked on before and what they were trained to do. And funding agencies will only fund you if you've already done most of the work. So it's very hard to totally change directions and take on something you've never done before. And we're lucky at the Wyss Institute is that we got a big philanthropic gift that gave us this incredible freedom. And the gift was actually explicitly not to do standard government-funded, iterative, incremental stuff, but to take on risks and do high risk, high impact. So that allowed us to really shift directions and take on lots of different opportunities. The other place that does it is when people are trying to solve a particular problem. Certainly, I think the SARP-H initiative is a great way to do that, because they're explicitly putting funding out that are carrots to scientists to change direction, and they don't have to have worked in that before. But if you were just to look, like, the system tends to work very incremental changes, and you're doing what you did before. So if the history was that it was mostly male-oriented focus, it's going to take a long time to change that, unless you start having additional funding out there that recognizes you don't have to have worked in this before, because by definition, if you did, you're limited to a very small number of people. Plus, those people are probably going to do what they did before, so you're not going to do, breakthrough new innovative stuff.
You could take any organ and virtually any problem and start to come at it with an organ chip. I mean, is that too broad, or is that right?
Pretty much. I mean, we haven't found one we couldn't do yet when asked to do it. So that's all I can say. I mean, the other thing I didn't say is that we actually have linked many of these together by the blood vessel channel, so that we transfer fluid from one to the other to create what we call the human body on chips. And so we did, I think, 8 organs. We've done up to 10 organs together. And, you could add a drug, watch it be metabolized by the liver, which, releases an active metabolite, and see how that acts on things. And you could see how different organs work together.
So when you think about sort of 5 years from now, do you think we'll have married organ on a chip with some areas of women's health where viewers and listeners see the direct benefit in their lives, or is it more longer term?
If this clinical trial works and we end up confirming that we could kind of replicate patient responses on these chips and then lead to better and better versions, that's something that could have impact in the next 5 years, because those trials are starting soon. We have other drugs that are in clinical trials for like viral infections and things like that. This last month, there was news around the world that the first kidneys from pigs that were engineered so they don't have viral infection were successfully transplanted into humans, and the humans left the hospital and are doing well. The people that get the credit were the surgeons who implanted the organ. But the technology was actually developed at Harvard and the Wyss Institute, and it went out as a company. Neither the company nor Harvard or the Wyss were mentioned. But it's also true about science in general. The public doesn't realize that the economy is driven by industry, which is driven by new technologies, which is driven by science, right? I mean, we wouldn't have microchips, optical fibers, drugs without the science. And that's where the big money makers are, at AI, but they don't see that as connected to scientific research.
Yeah, and what's fascinating is when you talk as the inaugural director of the Wyss Institute, with the largest gift ever given to Harvard in 2009 to start the Wyss Institute by Hans-Jörg Wyss, $125 million, you are uniquely positioned to, and you have, thank you for all that you've done, make major transformations when it comes to science. I think it's so hard. So many people think that the idea or the science is the key to bringing that translationally to humanity, but creating a business around a brilliant piece of science is, probably 90% of it. And I think that it's incredibly admirable, your focus on translation and realizing that there is that gap and helping to serve as a model for the world in terms of what translation could look like. I think that's what you and the Wyss Institute are doing.
It's not enough scientists realize this. I mean, I learned it early on because I got involved with things moving to the clinic and startups, but there are scientists who are ashamed to put in a patent. And I try to explain to them, it will never reach a patient. It will never help anyone if you don't patent it, because no one is going to put the investment you need to bring it all the way to patients. And as you said, the hardest part of the process is actually commercializing these things. And that's why we set up major focus of the Institute is developing a whole funnel and pipeline and mentorship and an innovative process to accelerate that. And it's worked really well. As you said, we've had something almost 60 startups. It's really incredible.
Well, congratulations on all the success. Thank you for being on the podcast. Thank you for everything you're doing in the world. I really enjoyed spending time getting to know you and just appreciate you and appreciate your time today.
No, it's always a pleasure talking, Sharon. Thank you so much.
Thanks. Thank you for tuning in. Please connect with me, Sharon Kedar, on LinkedIn for additional innovative content. If you enjoyed this episode, please take a moment to like it, and don't forget to subscribe to the channel by clicking the button below this video. The views and opinions of the host and podcast guests are their own professional opinions and may not represent the views of North Pond Ventures.